It is the chapter of gibberellins, BSc second year. Gibberellins were first discovered by Japanese farmer during 1800 in rice disease called Becken, which did not produce seeds. The disease was also caused by a fungus Gibberella fusicori. Gibberella fusicori. Also known as Fusarium uniformis. In 1926, K. Kurosawa found some chemical parasitizing this plant. Thereafter, in 1939, T. Yobita and T. Hashi achieved a crystalline material was known as gibberellin and was ultimately isolated from Gibberella. So that was the history of Gibberella and gibberellin. Structure. Gibberellins are almost of similar structures with a gibbon ring made up of a cyclohexane ring and a 4-lactone ring. They differ only in minute details into the number of and position of hydroxyl and sometimes alkyl and carboxylic groups at different carbon atoms. The given ring is there, which is complicated. G3 is usually called gibberellic acid. Extraction. Extraction of GX, it's 1, 2, 3 derivatives. The plant material is usually macerated with cold methanol containing 50% water. After evaporating of the organic solvent and sanctification, the aqueous residue is partitioned against ethyl acetate at different pH values. The GS are extracted into ethyl acetate at pH 2.5 to 3.0, re-extracted into buffer at pH 8.0 and again extracted into ethyl acetate after adjusting pH 2.5 to 3.0. Almost all free GS may be obtained in the final acetate, ethyl acetate, fructose. Chemical structure. Gibberellins are abbreviated GA1, GA2, GA3, etc. The original GA3 is gibberellic acid. They initiate elongation of stem and cell division. Elongation and cell division. It has 19 or 20 carbon skeleton derived from terpenoids and grouped in 4 or 5 rings. This skeleton is called as gibberellin. Gibberellin. A ring is different in all types. They all seem to have a carboxylic group, and because of this they are referred as gibberellic acids. Physiological effects. Promotion of plants growth. They promote growth. Ground growth means full growth in intact plants. It is a biological tool, biological engineering tool as a supplement. Number 2, GA enhances the elongation of the stem in intact plant much more than the excess plant stem segments. Therefore, genetic dwarfness is overcome. Genetic dwarfness is overcome. So it is The distinguished characteristic. Dwarfness is overcome in maize, bean, pea. Dwarfness is due to mutation that blocks biosynthesis of gibberellic. Enzymes are blocked somewhere. Third, promotion of bud growth and seed germination. GA promotes seed germination in several species which fail to germinate when dormancy is removed. They enhance cell elongation, so the radicle can push up through endosperm, seed coat, or fruit that restricts its growth. Subsequently, the hormone also stimulates hydrolysis and transport of stored food materials from endosperm and cotyledons to the growing root shoot axis. Here, sucrose and amino acids assist in germinating seeds. Sucrose and amino acids are important part to germinate seeds. Third, induction of flowering. Anton Long et al. 1957 reported flowering and many herbaceous plants. With the rise of GA level, exogenous application is applied to induce flowering in some photoperiod sensitive and cold requiring species. GA induces sex expression in plants. Cocomus sativa, cannabis, bryophyllum are examples to reproduce male flowers. Female flowers are reproduced in maize. Parthenocarpy is also developed. Parthenocarpy is also developed. Fourth, bolting. Bolting here the leaf growth is tremendous, whereas retardation in internodes is observed. Is rosette when there is a striking stimulation of internode elongation, and sometimes the internode elongates 5 to 6 times of the original height is known as bolting. So 5 to 6 times elongation is known as bolting. Five, prevention of senescence. Fleischer and Osborne 1965 observed that exogenous application of GA can prevent senescence of leaves. In the leaves of Tropiolum, endogenous GA content decreases along with senescence. In leaves, gibberellins are sprayed to avoid rind disorders, means peeling disorders in oranges, citrus, such many fruits. It delays senescence and maintains firm rinds, peels. Delicate firm peels are maintained, developed. Next, sixth, mobilization of foods and minerals in seeds. The organic food and minerals is stored in the form of macromolecules in the endosperm, and cotyledons are released and transported to the growing root and shoot axis until the seedling is established in the soil. GA is known as to stimulate the hydrolysis of stored macromolecules and their transport to the embryonic axis. GA germination and growth. Parthenocarpy. Formation of seedless fruits without pollination and fertilization is called parthenocarpy. It can be induced into tomato, potato, banana, orange, guava, etc. GA is characterized by many features. One, flowering and development of fruits. Number two, conversion of biennial to perennial plants. Third, will increase the number of fruits and size also. Fourth, to break dormancy that you have seen. Five, increasing photosynthetic and respiratory growth along with photoperiodic growth. Photoperiods are maintained and thereby the plant's growth takes place at a very good level. 6. Increasing cambial activity. Cambial activity means the cambium is activated tremendously here. Biosynthesis of GA. You see here, biosynthesis, it is outlined. This has been worked out in the mature seeds. Plastids are sites of biosynthesis. Plastids are here, or these sites of biosynthesis. Synthesis of linear chains of geranyl, geraniol, pyrophosphate from acetate. Number two, cyclization to form corine. So you see acetate to acetoacetyl coenzyme and this acetoacetyl coenzyme to mevalonic acid and isopentenyl pyrophosphate. This isopentenyl pyrophosphate to geraniol pyrophosphate, further farnesyl pyrophosphate, and further geraniol pyrophosphate. Then later on phosphon D that may inhibit, and lastly corine is formed. Corine is formed in the form of GA4, GA12, and the biosynthesis ends here. These steps involve oxidation and experimental work. So it is a very complicated process here to synthesize corine as GA4, GA12 in the form of the gibberellic acid. Commercial importance of GA. Horticultural and food industries use this GA. They are spread on vineyards, grapeyards to increase the number of and size of grapes in the cluster. Thompson seedless grapes are used with the significance of application of GA. Apples, pears, and many fruits are changed. They are changed by improving colors, size, quality. Bud formation and setting of fruits are stimulated. With this hormone, GS are used to increase alpha amylase activity. Alpha amylase activity. In germinating barley seeds to be used for malt production in beer industry, beer, wine industry, GA elongates internodes in sugar without changing or affecting sugar content. So it is a horticultural use of sugarcane and the industrial use of sugarcane with the quality and yield of sugar without changing its other contents and quality. So many such industrial uses are there. That's all.